Today we will discuss about one more new topic, ketone bodies. Now introduction about ketone bodies. Carbohydrates are essential for metabolism of fat, or fat is burned under the fire of carbohydrate. Acetyl-CoA formed from fatty acid can enter and get oxidized in DCA cycle. The condition is only when carbohydrates are available. During starvation and diabetic mellitus, acetyl-CoA takes the alternative route for formation of ketone bodies. There are three main ketone bodies. There are acetone, acetoacetate and beta hydroxybutyrate. These three are known as ketone bodies. It also known as acetone bodies. First two are known as true ketone bodies. These acetone and acetoacetate are known as true ketone bodies because it have that hetero group C double bond O here and here. The ketone functional group is there, so acetone and acetoacetate are considered as true ketone bodies. Acetone and beta hydroxybutyrate, they are synthesized from the acetoacetate. So acetoacetate, these both are synthesized from the acetoacetate. So acetoacetate is known as primary ketone bodies. Others are secondary ketone bodies. The acetoacetate is primary ketone bodies. These two are secondary ketone bodies. Beta hydroxybutyrate does not possess the keto group C double bond O. Acetone and acetoacetate are true ketone bodies. Ketone bodies are water soluble and energy yielding. Then properties of ketone bodies. It is water soluble substance. Plasma level of ketone bodies less than 1 mg per deciliter. Urine level of ketone bodies less than 3 mg per 24-hour urine. Plasma level ketone bodies less than 1 mg per deciliter. Urine level less than 3 mg per 24-hour urine. Acetone is synthesized in smaller amount and comparing to other ketone bodies because it cannot metabolized. Okay, it is a volatile substance that is excreted by the lungs. So that reason only acetone are synthesized from smaller amount and comparing to the other ketone bodies. Acetoacetate and beta hydroxybutyrate are relatively strong acid. Ketone bodies are detected in urine by the test name is known as Roth's test. By using Roth's test, we can detect only acetone and acetoacetate. Beta hydroxybutyrate we can detect it by modified Roth's test. Special test we are using for detect that acetoacetate, Gerhan test. These are the properties. It is a water soluble substance. Plasma level less than 1 mg per deciliter. Urine level of ketone bodies less than 3 mg per 24-hour urine. And comparing to other ketone bodies, acetone are synthesized in a very small amount because it not metabolized. It is a volatile substance excreted by the lungs. Acetoacetate and beta hydroxybutyrate are strong acid. Ketone bodies can detected by Roth's test. Beta hydroxybutyrate are detected by modified Roth's test. Special test used for acetoacetate detection, that name is called as Gerhan test. This is introduction, then properties of ketone bodies. Now coming to ketolysis. What is ketolysis? Utilization of ketone bodies. Where it will occur? Mitochondrial matrix of extrahepatic tissue, especially our kidney cortex, cardiac, skeletal muscles, which normally utilize the ketone bodies for source of energy. Our brain can also utilize the ketone bodies as a source of energy. Only the condition is prolonged starvation. They provided 75% of energy if they need. So what is ketolysis? Utilization of ketone bodies, where it will occur mitochondrial matrix of extrahepatic tissue like kidney, cortex, cardiac and skeletal muscles. Brain also utilizes the ketone bodies as a source of energy. The condition only during prolonged starvation. It provided 75% of energy. Ketolysis does not occur in RBC and liver. Why it does not occur in RBC means absence of mitochondrial matrix. Why ketolysis does not occur in liver means due to the absence of thioporase enzyme. This thioporase enzyme is essential for the activation of ketone bodies. So liver absence of thioporase enzyme. So ketolysis does not occur in RBC and liver. Ketolysis, utilization of ketone bodies, site mitochondrial matrix of hepatic tissue like kidney, cortex, cardiac and skeletal muscles. Brain also can utilize the. It does not occur in RBC and liver. Why RBC does not occur means absence of mitochondrial matrix. Why does not occur in liver means absence of thioporase enzyme. This enzyme required for activation of ketone bodies. Now moving to ketogenesis. What is ketogenesis? Formation of ketone bodies. Ketone bodies are synthesized only in liver. Ketogenesis, formation of ketone bodies. Ketone bodies are synthesized only in liver. Now synthesis in proper. 2 molecules of acetyl-CoA condense to form acetoacetyl-CoA. This reaction catalyzed by enzyme thiolase. 2 molecules of acetyl-CoA condense to form acetoacetyl-CoA with the help of enzyme thiolase. This acetoacetyl-CoA combined with another molecule of acetyl-CoA to produce beta-hydroxymethylglycerol-CoA. DHMG-CoA synthase catalyzing this reaction regulate the synthesis of ketone bodies. So it is a regulator enzyme. HMG-CoA synthase is a regulator enzyme. Again, HMG-CoA lyase cleave to produce the acetoacetate and acetyl-CoA. We already know this acetoacetate is primary ketone bodies. This lyase enzyme cleavage here it produced acetoacetate and acetyl-CoA. Again here decarboxylation and dehydrogenase process will occur. Acetoacetate entered to dehydrogenase process, it produced beta-hydroxybutyrate. Acetoacetate entered to decarboxylation process, it produced acetone. So these two are secondary ketone bodies. This one is the primary ketone body. Ketogenesis notes format. Formation of ketone bodies name is called ketogenesis. Ketone bodies are synthesized only in liver. First enzyme thiolase, second one is HMG-CoA synthase, Third one is HMG-CoA lyase. Here decarboxylation, dehydrogenation process will occur. Dehydrogenase process, it produced beta hydroxybutyrate. Decarboxylation process, it produced acetone. So acetoacetate. Two clinical condition ketone bodies are synthesized. First one is starvation, second one is diabetic mellitus. Fat become the source of energy and its degradation is greatly accelerated. That means increase in rate. It results in excessive production of acetyl-CoA which cannot be fully utilized through Krebs cycle. So, because lack of oxaloacetate, overproduction of acetyl-CoA cannot accepted by Krebs cycle because lack of oxaloacetate. Therefore, it will converted into ketone bodies. Two clinical condition: overproduction of ketone bodies, starvation and diabetic mellitus. Excess amount of acetyl-CoA cannot utilized by Krebs cycle because less amount of oxaloacetate, it cannot accepted that acetyl-CoA. So it will converted into ketone bodies. Now coming to biochemical and clinical finding. It will causes ketonemia, that means increases in ketone bodies. Then it causes ketonuria, increases ketone bodies in urine. These both condition together, ketonemia and ketonuria, is called as ketosis. Increase amount of ketonemia and ketonuria. This condition name is called as ketosis. Then it causes odor of the fruity odor of breath because of acetone. We already know acetone is excreted by the lungs, so it produced that odor in breath. Then it causes excessive accumulation of beta hydroxybutyrate and acetoacetate causes metabolic acidosis, then hyperkalemia. Acidosis result in the shift of potassium from intracellular to extracellular compartment. Because of acidosis, that potassium will shifted to intracellular to extracellular compartment. It causes hyperkalemia. Then it also causes dehydration, coma. If not treated, means it leads to death. So what are the condition? Ketonemia and ketonuria. These both condition together name is called as ketosis. Then odor of breath, acetone fruity odor, it causes metabolic acidosis, hyperkalemia. Also causes dehydration, coma. If not treated, means leads to death. How to management of that overproduction of ketone bodies? Treatment to give insulin and glucose. When glucose and insulin we will give to intravenously, that potassium is trapped within the cell. Then administration of bicarbonate and maintenance of electrolytes and fluid balance is very, very important. So treatment purpose will give insulin and potassium, then administration of bicarbonate. Biochemical and clinical finding. Management. Next, moving to important test, row thrust test. Ketone bodies are detected in urine by the test are known as row thrust test. By using this test, we can detect only acetone and acetoacetate. We already know beta hydroxybutyrate can detected by modified row thrust test. Plasma level of ketone bodies less than 1 mg per deciliter. Urinary level of ketone bodies less than 3 mg per 24-hour urine. Here we are using nitroprusside, sodium nitroprusside used in this test to react with acetone and acetoacetic acid. In the presence of alkali means it will produce the purple color. Then reagent, what are the reagent? We need ammonium sulfate crystal 1 gram, then 2% of sodium nitroprusside, then liquor ammonia. For detection of ketone bodies, what are the things we need? Ammonia sulfate crystal, 2% of sodium nitroprusside and liquor ammonia. Method, we will take one clean test tube. It containing 3 ml of urine. After that we are adding ammonium sulfate 1 gram, then 3 drops of sodium nitroprusside. Then we will shake well, then slowly add ammonia solution. Purple ring forms presence of ketone body in urine. Ketone bodies found in urine during two conditions, diabetic mellitus and starvation. We already know normal production of ketone bodies 1 mg, less than 1 mg per deciliter. So Rothrus test used to detect ketone bodies. What are the reagent we are using? Ammonium sulfate crystal, sodium nitroprusside and liquor ammonia. Test tube containing 3 ml of urine, then we are adding ammonium sulfate, then 3 drops of sodium nitroprusside, then we are adding ammonia. It produced pink color. Ring means positive, that means presence of ketone bodies in urine. Thanks for watching.